Ah, of course. Absolute zero. Oh, hello there. I didn't notice that you came in. Don't mind me, I was just reading some physics. Anyway, over the weekend, I acquired this plant. Don't know where I got it from, but I do know that this little guy can make his own food. How, you say? Through a process called photosynthesis. If I could direct your attention to the whiteboard here, right here we have the photosynthesis equation. Now this tells us what photosynthesis is, carbon dioxide, water, light energy, glucose, oxygen. That's what photosynthesis is. But what it really is how plants turn light energy into an energy that can be used readily by its cells. It takes place in two distinct phases, light-dependent reaction, and we have the light-independent reactions. Let's switch over to VoiceOver and we can look at the chloroplast, the organelle where this all takes place.
Right now we're switching over to voice commentary to look at the structure of a chloroplast. Now as you can see, just like in mitochondria, we got an outer membrane and an inner membrane of the chloroplast. But right here you see these little disc structures. These are called thylakoids. And when they're stacked in a row like that, they're called granum. Of course, you got the stroma, which is the fluid that surrounds the thylakoids, as well as the inside of them where you got the lumen. Now, the thylakoid membrane is actually where the light-dependent reaction is going to take place, and we're going to switch on over and look at that right now.
To start the light-dependent reactions, first, photosystem 2 absorbs photons of light, exciting electrons to be passed down the photosynthetic electron transport system. Electrons lost are replaced by the oxidation of water, producing oxygen gas. Next, the electrons flow through the B6F complex toward photosystem 1, providing energy to pump protons into the thylakoid. Then down the ETS, photosystem 1 absorbs more photons in order to reduce NADP plus to NADPH. These electrons are replaced by electron transport from photosystem 2. As the excited electrons flow through the ETS, a proton gradient is created. The protons then flow through ATP synthase, causing ADP to bond with a high-energy phosphate, making ATP. The NADPH and ATP are now ready to be used in the light-dependent reaction which takes place in the stroma. The light-independent reactions, or the Calvin cycle, can be broken up into three basic phases. In phase 1, carbon fixation, Rubisco turns 6 molecules of carbon dioxide into 12 molecules of 3-phosphoglycerate. In phase 2, reduction, energy from ATP and NADPH are used to turn 12 molecules of 3-phosphoglycerate into 12 molecules of glyceraldehyde 3-phosphate, 2 of which go to glucose, 10 of which go to phase 3. In phase 3, regeneration of RUBP, ATP is used to turn the 10 molecules of glyceraldehyde 3-phosphate into 6 molecules of RUBP to be used in phase 1. Since one turn of the Calvin cycle only yields 2 molecules of glyceraldehyde 3-phosphate, the Calvin cycle needs to turn many times to get enough glyceraldehyde 3-phosphate to make glucose and other sugars. In short, photosynthesis uses light energy and H2O to make NADPH and ATP, which are then used to turn inorganic CO2 into organic carbohydrates with energy-rich CH bonds, ready to be used by the cell. What are you still doing here? The video's over. Citations in the description box below. Go home. Live your life.